Today we start a new chapter, chapter 7, and we're looking at hemoglobin. The objectives are here on the board. We're going to be looking at the structure of hemoglobin and how that relates to its function. And we'll be looking at how different organisms have different hemoglobins and why that is. And we'll be looking at some of the terminology involved with hemoglobin, because once we've got that terminology, we're a step closer to passing that exam. A quick recap of what do we mean by primary structure, secondary, tertiary and quaternary. So of course your primary structure is how those amino acids order themselves, it's the sequence. And the secondary structure is, in this case with hemoglobin, how they coil, they enter an alpha helix. The tertiary structure is how that alpha helix then bends upon itself and all the hydrogen bonds that are made with that. And the quaternary structure is when you've got more than one polypeptide chain interacting with each other and they come together. So two or more polypeptide chains together is what we would consider to be the quaternary structure. Now, in the case of hemoglobin, it's made of 4 polypeptide chains, and 2 of them are called alpha helixes or alpha chains, and 2 of them are called beta chains, and you can see those here. This is an old image from AQA, and what you'll see in here is you've got your alpha chains, here you've got your beta chains, and what each of these chains has in the middle of it is something called a heme group, and that is the prosthetic, the false bit, which is made of iron. So each of those holds a molecule of iron, and it's the iron that bonds to the oxygen. So depending on how exposed that molecule is, depends on how easy the oxygen can bind. We'll come back to that later. So in a nutshell, what we're going to be learning is how when one oxygen molecule bonds, it causes a change in the shape that then changes the shape of the second and the third and the fourth, and the binding of oxygen to each of those polypeptide chains is a little bit different. So when we talk about hemoglobin, we talk about its affinity for oxygen, its liking for oxygen. And when we talk about oxygen being taken in by the molecule, we talk about its association with oxygen or its loading with oxygen. And likewise, it unloads or it dissociates when it lets that oxygen go. Before, we've always just talked about red blood cells pick up oxygen at the lungs and transfer it to respiring cells. But what we're really thinking about now is, what is it that tells it to pick it up, and what is it that tells the hemoglobin to let it go? And clearly, if we're just thinking about that in everyday terms, then we can think, well, actually, yes, when there's a high level of oxygen, we want hemoglobin to associate, and when there's a low level of oxygen, perhaps at respiring cells, we want it to dissociate, we want it to let it go. So that gives us a little bit of a clue of how things are going to go as we progress through the lesson. So here's a worksheet I'll be asking my students to do, and what I want is a definition or a commentary, some statement that explains each of these. So let's have a little look. First things first, loading. Well, when the hemoglobin molecule loads with oxygen, it's accepting oxygen into its molecule and it's going to carry it around. So loading or associating, same, that's when the hemoglobin takes up or takes on oxygen. When we look at unloading or dissociating, we're talking about that hemoglobin molecule lets go of the oxygen into the surrounding tissues. A hemoglobin with a high affinity. Now let's think about that for a minute. A hemoglobin with a high affinity, a high liking for oxygen. That means it's going to bind really easily. So in low oxygen levels, it's going to bind onto that oxygen. But it equally means that when it gets to low oxygen levels, it's not as easy to let it go. So hemoglobin with a high affinity binds easily, it associates easily, but it dissociates less easily. And we can also say the same is true. A hemoglobin with a low affinity for oxygen, let's think about that for a minute. It's got a low liking for it. So oxygen levels have to be really quite high before hemoglobin will bind with that oxygen. What that actually means is that in low levels of oxygen, it's quite happy to dump that hemoglobin too, because it's got a low liking for it. If we look at the importance of hemoglobin, we always need to consider the importance of processes and substances, proteins, in the human body. The reason why is because in year 13 you have to do an essay, and the essays are always titled The Importance of. When we're thinking about the importance of, what we think about is what would happen if we didn't have that particular process or that particular molecule. So what would happen if we didn't have hemoglobin? Well, what would happen is we wouldn't have a transport molecule to transport it deep inside of our bodies. We would be relying on diffusion from the outside, and in that case we would need a massive surface area compared to our volume. Human hemoglobin has an additional importance, and the additional importance is due to the fact that its affinity changes for oxygen depending on the partial pressure of oxygen in the air. So as there is less oxygen, its affinity changes compared to when there is more oxygen, and that is key later on. The next few things on this list we'll just quickly talk about, but we'll learn about them in more detail later on. So the next one says, what is it about its structure that enables it to have a changing affinity? Because as one polypeptide chain bonds with an oxygen, it then further exposes the second, third and fourth polypeptide chain to accepting the oxygen more easily. So you don't need so much of a change in the partial pressure of oxygen for more rapid binding to take place, and that's called positive cooperativity, because one binding has caused a change in the affinity of the second, third and fourth polypeptide chain for oxygen. The next one says, how does adding carbon dioxide affect the affinity of hemoglobin to carbon dioxide? Well, when you add carbon dioxide, you reduce the affinity of hemoglobin for oxygen. Let's think about that for a moment. If you are at respiring cells and you want your hemoglobin to dump your oxygen, well, one way of doing that is by increasing the levels of carbon dioxide, because that is what would happen if the cells were actively respiring, and it's going to be more able to dump the carbon dioxide. So when you raise carbon dioxide levels, the affinity of hemoglobin for oxygen will change. It becomes less, has a less affinity for oxygen, so it's more likely to dump it. So if we were looking at this on a curve, it would move the curve to the right if partial pressure was along the bottom, and we'll look at this in a moment and explain it in a little bit more detail, but this is just basically it in a nutshell. Increased carbon dioxide moves the curve to the right because it reduces the affinity of hemoglobin for oxygen. And the last one says, different organisms have different hemoglobins, i.e. they have a different amino acid sequence. And the reason why is because they live in different partial pressures of oxygen. Consider a worm that lives under the ground. It might live in largely anaerobic conditions, but it still needs oxygen for its respiration. So it will need a different level of affinity. How does it change that? It changes it by its amino acid sequence. We're looking now at a graph that represents a sample of hemoglobin. We're not looking at one hemoglobin molecule, we're looking at hemoglobin in a sample of blood, for example. Along the bottom, we've got the partial pressure of oxygen. That just represents how much oxygen is in the air. So at a low partial pressure of oxygen, there's not much of oxygen available in the blood plasma or the air, whichever you like to think about it. And at higher, there's a lot more oxygen available. So it's quite self-explanatory if you're not a chemist. Up the side, we've got the idea of percentage saturation of hemoglobin of a sample, not just one molecule. So what does this curve show? It shows that over a large increase in partial pressure of oxygen, there isn't that much increase in its saturation. Then, over a very short distance of increase of partial pressure, there's a massive change in the percentage saturation of hemoglobin, and then it levels off. It doesn't matter how much more we increase the concentration of oxygen around the hemoglobin, there's very little change. So that's what the graph shows. So what do we need to gather from this graph? Remember, there are 4 polypeptide chains. Within each chain, there is a prosthetic group, a binding site, and they are hidden at different points. And what basically happens is, as we increase the partial pressure a lot, that first binding site of one of the heme groups will then accept an oxygen. That oxygen atom then, combined with the heme group, changes the structure of the second, the third and the fourth polypeptide chain. That means then that over a very small increase in partial pressure, oxygen can bind to number 2, number 3 and number 4 polypeptide chain. However, actually what happens for the fourth one is that the rate of binding slows down, its affinity is decreased, and that's just because most of the groups, the 3 out of the 4 binding sites, are now taken up with oxygen, and the chance of the oxygen atom just coming along and hitting that 4th binding site is now reduced. So what we see is a levelling off of the affinity for oxygen there. So remember, we're not just looking at one red blood cell, one hemoglobin molecule, we're looking at a sample of hemoglobin, and that's how we get this nice curve. So I'll just go over it again. The partial pressure increases to the point then that one of the polypeptide chains will bind. That changes the shape of the second, third and the fourth, and so that over a small increase in partial pressure, a lot more oxygen can bind. And that cooperation between the heme groups is called positive cooperativity. So one helps the other, which helps the other, which helps the other. And remember, we said that the only reason why it tails off for the 4th heme group isn't that it isn't exposed, it's just that most of the binding sites are taken up. The concentration, the partial pressure of oxygen is therefore lower, if you like, because there's just less chance that it will then bind. And that's all I want to cover in this lesson before we move on to 7.2, where we start looking at how this curve will shift to the left and to the right, or indeed how it is shaped differently for different organisms that live in different partial pressures of oxygen. Here's a worksheet I'd like my students to do. It just basically relates the exposure of the binding sites of each polypeptide chain of the hemoglobin molecule to the dissociation curve. That's the curve that we were looking at of partial pressure of oxygen against percentage saturation of hemoglobin. In this first one, you can see that the binding site of each polypeptide chain are obscured, they're hidden. One of them is slightly available, and as the partial pressure of oxygen increases, this one can bind in here. And we can see that there. On binding, it then changes the interaction of the quaternary structure of the hemoglobin molecule, and more binding sites on the other polypeptide chains become exposed. They take on the second and the third really quite readily, but they leave the fourth not filled in, because as the binding sites are filled, the chances of an oxygen molecule coming along and fitting in there are reduced, and so that's what gives us the characteristic S-shaped curve.